It is my pleasure to introduce our Grand Round speaker today, Dr. Gordana Raca, who will be talking to us from Los Angeles. I guess one of the downsides of Zoom is that she had to get up very early to actually give this talk. But one of the upsides is that in Minneapolis right now, it's 28 degrees and we look to a high of 48. And for you, I think it's 70 now with a high of 92 for today. So there are pluses and minuses of both. By way of background, Dr. Raca received her MD degree from the University of Novi Sad School of Medicine in Yugoslavia. She then completed a master's degree in cytogenetics at the University of Belgrade, Yugoslavia, followed by a PhD in molecular genetics at the University of Chicago, and then 2 fellowships, one in molecular diagnostics at the University of Chicago, and a subsequent fellowship in clinical cytogenetics at Emory University. And she is boarded in both of those disciplines. Dr. Raca served on the faculty of the Department of Pathology and Laboratory Medicine at the University of Wisconsin-Madison, and then in the Department of Medicine at the University of Chicago. But in 2015, she was lured away from the University of Chicago by Dr. Jacqueline Beagle to accept a position at the Keck School of Medicine, where Dr. Beagle and a small but very talented group of faculty developed the Center for Personalized Medicine at Children's Hospital of Los Angeles. As you will hear, their development and continued evolution and integration of genomic technologies is remarkable, and Dr. Raca serves as professor and director of clinical cytogenomics. Although the integration of cytogenetics and molecular genetics is now recognized as essential for laboratory genetics, few, if any, individuals have the depth of knowledge in both disciplines that Dr. Raca has. As a result, she has been propelled to leadership positions on almost every one of the highly regarded national committees that are tasked with setting practice guidelines and working with the interpretation of germline and somatic variants in hematologic malignancies and solid tumors. I'm personally very honored to partner with her in the genomic risk assessment of pediatric AML and children's oncology group. She's a wonderful team and individual scientist, and her contributions to the field, as documented in her publications and committee work, are invaluable for advancing knowledge in both clinical and basic scientific domains. So thank you very much for coming, so to speak, Gordana. And today she will speak about the utility of RNA sequencing and optical genome mapping for comprehensive assessment of genomic abnormalities in pediatric B-lymphoblastic leukemia.
Thank you so much, Betsy. Thank you for that really kind introduction. It is a great honor and pleasure to be joining you this morning. And I'm thrilled to have the opportunity to tell you about our work. And so, as Betsy mentioned, I will be talking today about using RNA sequencing and optical genome mapping for comprehensive assessment of abnormalities in genetic abnormalities in pediatric B-lymphoblastic leukemia. And before I start, I do want to acknowledge my colleagues and collaborators who worked with me on these studies. So why do we pay so much attention to testing, genetic testing for BLL at our institution? We're a pediatric hospital, and BLL is the most common pediatric malignancy. It accounts for 25% of cancer diagnosis in children up to 15 years of age. And that also means that it is one of our high volume malignancies that accounts for a high volume of our testing. Accounts for about 1/3 of our cancer samples, with the second third being all solid tumors combined and the third being all brain tumors combined. So it's a high volume testing for us to test BLL for genetic abnormalities. Also, genetic abnormalities are very important in risk stratification and management of patients. For decades now, we have been using cytogenetic and FISH abnormalities, and in modern era, genetic abnormalities detected by molecular methods in risk stratification for patients with BLL. And then also it is a pretty challenging testing for clinical laboratories. BLL is a very heterogeneous disease. Main drivers belong to different types of genetic abnormalities, including abnormal gene fusions, copy number abnormalities, and sequence variants. So it's difficult to find one integrated test that has good performance in detecting these disparate types of genetic changes. And then finally, it's very extensively studied disease. Thousands of samples and in this modern genomic era have been studied by genome sequencing, RNA sequencing, advanced informatics analyses, and it is very difficult to keep up with this overwhelming amount of genetic information that we have these days for this disease. And just to illustrate this last point, if we look at genetic categorization of BLL in pre-genomic era in early 2000s, where we were only using cytogenetics and FISH to test these diseases, we knew about 7 different cytogenetic subtypes. And there were more than 25% of the cases that remained unclassified. And I have to say, sometimes I miss those good old days when everything was much simpler. Because when we compare that with this more recent paper where they analyzed more than 1,400 BLL cases, and here we're seeing their figure where they classified them by expression profile, and they were able to divide them into 24 different molecular subtypes. So things became much more complicated in terms of understanding genomic of this disease, and that this is not only relevant in research setting, because we also see acceleration in transitioning this knowledge from research into clinical use. For example, the most recent 2022 update in WHO and ICC classification of hematologic malignancies will be defining some of these new genetic subtypes as new entities in diagnostics. So how are we supposed to keep up with all this in a clinical setting when we perform diagnostic testing for BLL? So the way we initially tried to keep up with genetic testing for BLL at CHLA was to just keep adding more and more tests. So we still routinely do cytogenetics and FISH for every case. But then when we validated chromosomal microarray for detection of copy number abnormality genome-wide in tumor samples, we added that to our BLL testing. And then when we validated our custom NGS panel for pediatric malignancies, we added that as well to routine testing for all newly diagnosed and relapse cases. So that now our routine testing for every new case and relapse case includes karyotype, a pretty large FISH panel, chromosomal microarrays, and our NGS panel that we call OncoKits. So obviously this is pretty resource intensive, and it takes a pretty long time to complete. We have pretty long turnaround time due to this. And so we wanted to see, was it effective? Were we able to resolve genetic drivers, detect genetic drivers and resolve genetic subtype for our cases as we would like to. So we looked at diagnostic yield with this approach, and we did analysis twice. First with 160 BLL cases that were tested by all 4 modalities, and then more recently on 356 cases. And in both times, we had approximately 15 cases that stayed unknown after all our clinical assays. And I should say that in a genomic study that I showed on the previous slide, they were able to characterize drivers and find genetic subtype in 98%, so only 2% of the cases these days, if you do comprehensive genomic assessment, and stay unclassified and unknown in terms of the driver. So we still have a gap to fill. we still are unable to find driver in some cases that should be, based on current knowledge, classifiable. So we were wondering what could be the next step. Of course, we could continue adding even more and more tests, but clearly that would not be very effective. So we decided to ask the question, can we find a method and an assay that as a single assay will be effective in and have very nice high yield in detecting subtypes and genetic drivers in our BLL cases. And that's how we decided to evaluate optical genome mapping and RNA sequencing as tools for comprehensive detection of drivers in BLL. And for this, we looked at our retrospective cohort of 359 BLL cases, identified all the cases that remained unknown for which we had material. So that was 50 unknown cases. And then also we were using selected subsets of known cases, and we tested those samples and those cases using optical mapping and RNA sequencing. So I will start by describing our experience with optical mapping, but I would first like to just very briefly introduce this technology. So optical mapping is an assay that allows you to detect structural abnormalities in the genome, and that includes both copy number changes and balanced rearrangements, and it has very high resolution. So it's being presented as a method that can detect all the abnormalities that we currently detect by cytogenetics, so by karyotyping, by FISH and chromosomal microarrays. So it could potentially be a substitute for all these three types of assays. The way it works is that you have to start by isolating high molecular weight DNA, so it requires fresh material for DNA isolation. And then you do labeling with fluorescent tags that get attached to a 6 base pair palindromic sequence that's present with high density in the genome. You counterstain the rest of your DNA molecule, and then you stretch these molecules. You move them by electrophoresis through very narrow nanochannels that stretches the molecules so that you can image the pattern of the labeling pattern on every long molecule. And then you can overlap molecules, assemble them into contigs, and you compare the pattern of labels between a reference and your test sample. And based on changes in these patterns of labels, you can detect structural changes in your test sample. And so here is a figure that depicts how that works. And if you compare these labels between a reference in green and your test sample in blue, you can see changes in labels, and that will indicate where there's a structural change. Here's an example of a deletion in a test sample, expansion of a repeat array, translocation, insertion. So you can detect both balanced and unbalanced copy number changes. Small copy number changes, deletions, and insertions are detected by looking at labeling patterns or these fluorescent marks. And for large copy number changes, for example, whole chromosome gain or loss, they will detect them based on depth of coverage. So you also get this genome-wide copy number profile from this analysis. So we started our evaluation of optical genome mapping by testing some known cases. And here I'm showing results for a small set, only 17 cases. We tested more in the meantime. But the point was that the type that was detected by our clinical testing, by our standard of care testing, and optical mapping was concordant in all our cases. And although this is only 17, but we here have representation of subtypes that are driven by different types of abnormalities. We have some BLLs that are copy number driven, some that have abnormal fusions, some that have translocations that result in enhancer hijacking, and all of these were very easily detected by optical mapping. And here are just some examples. for example, this hyperdiploid case, you see that the pattern of chromosome gains detected by optical mapping matches very nicely with what we saw by array. Here is a case of a BLL driven by intrachromosomal amplification of chromosome 21, IM21, where you expect to see complex copy number abnormalities on chromosome 21, and that was clearly visible by optical mapping. And here we have an example of very small deletion, 47 kb, with an ERK gene easily detected by optical mapping. And then cases of doubled hypodiploidy, where you need a SNP pattern to see SNP pattern abnormalities to detect them by array, were also picked up. So we see abnormal SNP patterns by array and very similar patterns by optical mapping. Although SNP analysis is not really a strength of this technology, but in many cases, it still can be done. And similarly, if you looked at the cases that were driven by translocations that create gene fusions, they were all also very easily picked up by optical mapping. And so you see evidence of translocations, and then you can detect exactly the genes that were present at the breakpoints. So this was very encouraging when it came to known cases. And so we were also interested, can we have some additional diagnostic yield for our unknown cases? And I mentioned that we had 50 unknown cases, but unfortunately with optical mapping, we were only able to get results for 32 because you do need fresh samples to isolate large molecular weight DNA. So it's not always ideal for retrospective studies unless you're very systematically banking your samples. But among those 32 samples that we did test by optical mapping, we had 10 cases where optical mapping detected abnormalities, mostly abnormal fusions, and that were missed by standard of care testing for a variety of reasons, either because they are rare and were not known at the time when the testing was done, but we also had 4 cases where optical mapping detected completely novel fusions that have not been described in the literature for in BLL at the time of testing. So we had very good concordance for known cases and some additional diagnostic yield for unknown cases. As part of evaluation of optical mapping, we also decided whether to test if we can use it to characterize very unusual abnormality in BLL that we got interested in. And it's known as PAX5 intergenic amplification, or we like to refer to it as PAX5 intergenic tandem multiplication. So this is the case that brought this abnormality to our attention. It was BLL, standard risk BLL patient. And in leukemia sample, with our standard of care, with our usual testing, we couldn't detect any of the known genetic drivers. There were very few abnormalities detected overall, but by array, we saw this small copy number gain. There appeared to be 2 extra copies. And it was occurring in the 5' region of the PAX5 gene, and PAX5 is an important transcription factor for BLL development. So, important gene for for B lymphocyte development. So it's an important transcription factor in B lineage development, but also in BLL. And it does have abnormalities of PAX5 are important in BLL oncogenesis. And so we got interested, as I said, in this small copy number gain and start looking into its possible clinical significance. And it turned out that this abnormality had been described previously as a recurrent change in BLL. It was studied by several European groups. They used MLPA, multiplex ligation dependent probe amplification, a method that detects copy number changes in genes. And they described that the incidence of this abnormality in BLL overall was about 1%, 3% if you looked at BLLs that did not have known genetic drivers. And they consistently saw that the gain involved exons 2 to 5 of PAX5 gene, and these are exons that code for DNA binding domain. And they showed by FISH that extra copies were within PAX5 gene. So this was not insertion somewhere else in the genome. This was gained in in situ. And they described that this was exclusive with other major drivers. And so you did not see this in VLLs that had one of the known subtypes at the time. So they hypothesized that this can be associated with new molecular subtype of VLL. So in the meantime, after these studies were published, a new genetic subtype of BLL was described that is exactly characterized by abnormalities of PAX5. It's called PAX5-ALT, and it shows presence of a variety of abnormalities of PAX5 that are believed to be main drivers for this subtype. And in some of these PAX5-ALT cases, they were able to detect this PAX5 intergenic tandem multiplication. So now it's believed that this PAX5 intergenic amplification or multiplication is one of the genetic abnormalities characteristic for PAX5-ALT BLL. But when we were collecting this information about this abnormality, we realized that there are many things that are still unknown about it, and that nobody directly and accurately determined what is exactly a position of these extra copies relative to the rest of the gene in the genome. What is the orientation of these extra copies? How many copies you have exactly? And so we hypothesized that optical mapping actually could be a very good tool to look into this, because it is expected to allow direct visualization of the extra copies of this gained region in long molecules. So we decided to test if by optical mapping we can characterize this PAX5 intergenic tandem multiplications. We went back to in our cohort of 359 BLLs, looked for additional cases with this copy number gain. We were able to find 6 cases, and here's a screenshot showing how that gain looks by chromosomal microarray. So you see that gain is very similar in size, and it always overlaps 5' region of PAX5 and seems to include exons 2 to 5. And by array, it appeared that there were 2 extra copies of this affected region. So out of these 6 cases, we unfortunately had fresh material for optical mapping only for 3, but for these 3 cases, we were able to get optical mapping results, and I will show you how they look. So here's the first case, and we are zooming into a PAX5 region, and this light blue arrow is showing the original copy of the affected region, but then we were surprised to see evidence for 4 more copies. So we see the pattern that's repeating 4 more times. So there are 4 more copies of the region that are all in tandem orientation, in direct orientation and in tandem, and here they're visualized in the long molecules. So in this first case, there were 4 extra copies. In the second case, there were actually 5 extra copies of the affected region, again in direct orientation in tandem. And then in the third case that worked for us, we again see 4 extra copies of the region, and they're all in tandem in direct orientation. So with optical mapping, what we saw is that there are either 4 or 5 extra copies of this gain, always in tandem and in direct orientation, which was surprising because based on CMA, we expected 2 extra copies. And so we realized that there was underestimation of copy number in these cases by CMA and both by previous MLPA-based studies. So, of course, these results seem to open more questions that they answer, because now it becomes very interesting if we could look at more cases, what would be the range of copy number that you have in this amplified region? What is the significance of always having multiple extra copies, whether that's related to the mechanism, how these gains arise in the genome, in the chromosome, or maybe you need multiple extra copies for this abnormality to be oncogenic. And most interesting question, how this affects PAX5 function and downstream regulation, because I mentioned that this affects the region of the gene that codes for DNA binding domain. And it has been shown that these are in frame. And so you probably have a protein that has multiple copies of the binding domain, and that probably affects the binding to the targets and regulation of PAX5 target genes. And so it will be very interesting to study and understand how this leads to oncogenesis. But for our purpose, for our study, it was important that this showed us the ability of optical genome mapping to study repetitive regions and to resolve abnormal chromosome structures. So in conclusions for optical genome mapping part of the talk, based on our experience so far, we do think that optical mapping has potential to improve BLL diagnostics, because in our hands, it was detecting abnormalities that we detected previously by karyotype, by FISH and CMA. So using just one assay for all these types of abnormalities would improve our efficiency. It did identify drivers that were missed by other methods, so we could potentially have higher diagnostic yield, and that also it will allow us to more accurately determine structure of chromosome aberrations. Of course, it does have some limitations. It does require special DNA isolation to get long molecules, so you need fresh material. There are some subtypes of BLL that cannot be detected. That includes DUX4 rearranged and EPOR rearranged cases, because these are caused by very small insertions into IGH region. And IGH is very repetitive and complex region, and the insertions are very small, so optical mapping cannot map this. Breakpoints in repetitive regions may remain undetected. And then, of course, this is not a sequencing test. So if you have to detect sequence variants, then this will not be sufficient. So now I will switch gears and describe our experience with using RNA sequencing as a comprehensive test for BLL. So this slide is depicting our RNA-seq assay that we developed and validated for detection of abnormal gene fusions. So this was not specifically detected for developed for BLL. It was developed for all our tumor samples and cases, and as a new method to detect fusions on top of our NGS panel, and because our NGS panel is targeted, so only detects fusions that we are testing for, and for cases that remain unknown after that panel, we wanted to be able to offer more comprehensive fusion detection, and that's why we developed and validated this test in the lab. And so we use a little unusual library preparation method for RNA sequencing. We do enrichment for mRNA species using exome capture probes, and that allows you to enrich for all mRNAs, including hybrid mRNAs if you have an abnormal fusion. And by using enrichment with exome probes, we think that we increase our sensitivity for detection of abnormal fusions that are expressed at very low level. Then those libraries are sequenced by either NextSeq or HiSeq to get 100 million reads. And then fusions are detected by combination of different fusion colors, and using the pipeline that was custom developed by our bioinformatics team, and combines these 4 different fusion colors. So during validation, of course, we tested a large number of BLL cases for which we know that had abnormal gene fusions, and all those fusions were very easily detected by this new RNA-seq fusion assay. But then we realized that we could be using this assay in BLL for much more than just detecting abnormal fusions. Because as I mentioned at the beginning, new genetic subtypes of BLL were often defined and detected exactly based on their expression pattern. So we could potentially use RNA-seq data to detect expression pattern and detect genetic subtype that way for our BLL cases. This blue dot is supposed to indicate that all these different genetic subtypes you can actually detect from RNA-seq just based on their expression profile. But what's also exciting about RNA-seq is that many genetic drivers, many abnormal variants that drive these subtypes can directly be detected as well from RNA-seq data. Obviously, you can directly detect fusions. Interestingly, you can also detect copy number for large copy number changes. So if you can have whole chromosome gains and losses, you can actually pick that up from RNA-seq data. You can even detect sequence variants if they are expressed mutations, if they're present in RNA. And then there are BLL subtypes that are at this point only detected or only defined by their expression profile. So examples of that are ETV6-RUNX1-like, PEX5-ALT, KMT2A-like. These are BLLs that have expression profile that's similar to ETV6-RUNX1 positive, KMT2A positive, but they don't have these abnormalities. And we don't know at this point what are specific drivers for these subtypes, so we can only detect them at this point based on their expression profile. So RNA-seq is then the only assay that at this point can detect these subtypes, which means that potentially this is more comprehensive than any other test when it comes to genetic subtyping of BLL. So that's why we decided to test if we can use data from our RNA-seq fusion assay for more comprehensive evaluation of BLL cases. And to do this, we developed collaboration with Dr. Zhao Gu from City of Hope. He is now faculty at City of Hope here in LA, but before joining City of Hope, he worked as a postdoc at St. Jude's and Charles Mulligan Lab. And he was in that position one of the key investigators in the large-scale studies that used expression profiling and other RNA-seq analysis methods to detect novel genetic subtypes of the BLL. He was involved in detection of F2D subtype, a PEX5-ALT subtype. So his specialty is exactly this comprehensive RNA-seq analysis in BLL. So we worked with Dr. Gu to do a comprehensive analysis of RNA-seq data for our BLL cases. So again, we started with a set of known cases where we knew what the subtype was based on our clinical testing. And we tested 28 cases, and they were all fully concordant. The subtype determined by RNA-seq, by blindly doing analysis using Dr. Gu's pipeline, was exactly concordant with what we know the subtype was by clinical testing. And so for all these variety of subtypes, RNA-seq analysis easily detected and classified them correctly. And then we also looked at our 50 unknown cases. Here we were able to get results for all 50 cases, and of 50, 39 were classified successfully by RNA-seq analysis, so for 78%. So very promising that you would be able to increase your diagnostic yield with this type of analysis. So many of these new cases that were previously unknown ended up being fusion-driven subtypes. And here we see how you can detect subtype by expression profile, but you also can detect abnormal fusion. So you have two ways of confirming your subtyping. We detected not only some rare fusions that were missed by our diagnostic testing, but in 4 cases, we detected previously unknown unpublished fusions. And those were the same ones that we also detected by optical mapping. And then other subtypes that were detected in our unknown cases were a variety. And for some of them, we were also able to directly detect abnormal driver, the variant that was the key driver, for example, and these PAX5 missense mutations that define specific subtypes could be detected in RNA-seq data. EPOR overexpression could also be independently confirmed, but for some of the others, we could only detect them based on expression profile, for example, this ETV6-RUNX1-like or PAX5 alt subtype. And then 11 cases remained unclassified, but I do want to mention that many of those had relatively low blast percentage. Some of them even were post-transplant, so we still had residual chimerism, and that, of course, will limit your power to do RNA-seq analysis classification based on RNA-seq analysis and expression profile. So here I want to show how you can detect some of genetic variants that serve as a driver directly from RNA-seq data. Of course, obviously, you can directly detect fusions, but interestingly, you can also detect a large copy number abnormalities, and it's definitely whole chromosome scale copy number abnormalities. So here's an example of hyperdiploid BLL, and you see how it clusters with other hyperdiploid cases based on its expression profile. But you can also see evidence for chromosome gains, and the chromosomes that they were called as being gained in this sample exactly matched what we knew based on karyotype analysis. And you also can detect sequence variants if they are expressed. So a hotspot gain of function variants are often expressed and present in your mRNA, and you can detect them from RNA-seq data analysis. And we had pretty good concordance between variants detected by RNA-seq and by our NGS panel. And then there is really a lot of other data that you can gain, you can get from RNA-seq. And this is an example of looking at ethnicity SNPs. This is something that we are not considering for clinical testing, but can be very useful for our research studies. And so, but looking at ethnically specific SNPs, you can decipher ethnicity of your patients, and we had very nice concordance between self-reported ethnicity and what was predicted from RNA-seq data. And there is a lot of other information that you can gain from RNA-seq. And some groups have published detecting subset of Icarus deletions, the ones that that result in dominant negative form of Icarus and are expressed, mRNA is expressed, those can be detected. And Icarus deletions are important prognostic markers. You can also detect IgH and T cell receptor sequence of your abnormal clone so that you can use it later for MRD analysis. And so there is other type of information. It is really fascinating, the amount of information that you can gain from looking at RNA-seq data. And we are not the only group that thinks how RNA-seq analysis can have large utility in diagnostics of BLL. And here are just some examples that this type of approach is already being investigated and slowly moving into use. And so the two studies on the top are retrospective studies, but I really want to point out the two at the bottom. So these are examples of using RNA-seq analysis, a comprehensive RNA-seq analysis for genetic and genomic characterization of BLL cases in the context of clinical trials. And so for these two trials, one was for a Malaysia-Singapore group and the other from Dana-Farber consortium, and they're using RNA-seq analysis as their primary genetic analysis method and approach prospectively to classify their stratify their BLL cases for the trial. So it's something that's definitely implementable and in clinical and has can be used clinically or for either in diagnostics in individual labs or for clinical trials. So at the end, I would just like to finish by showing some case examples, starting with a couple of cases where we had combined optical genome mapping and RNA-seq data. And it was very interesting to put it all together. And I think that there was very utility in having complementary information from these two technologies. So I mentioned that in 4 of our cases, we discovered fusions that have not been previously reported in BLL. And for those, we had both optical genome mapping and RNA-seq data. So here's the first case. It was a standard risk BLL case and a 1-year-old. Clinical testing did not detect the genetic driver. And in this case, we had from optical mapping evidence of a translocation between chromosomes 9 and 13 with breakpoints in JAK2 and this novel gene RBM26. So we expected that there will be a JAK2 fusion. But it was really important to have evidence from RNA-seq that the fusion was indeed present and that it was expressed. And also we could see from RNA-seq that the fusion was in frame and that entire JAK2 kinase domain was present. So it had all the characteristics that you would expect from an oncogenic driver fusion. And we also know that JAK2 fusions in BLL are associated with so-called pH-like BLL subtype. So we expected that this is supposed to be pH-like in BLL, and indeed it clustered together with other pH and pH-like cases. And so by having both optical mapping and RNA-seq data, we were able to show evidence of a translocation on DNA level. We had evidence that the fusion was expressed, and then we had evidence that it was a key driver for this leukemia because it classified as pH-like as you would expect from a leukemia that has JAK2 fusion. And what was interesting about this case is that a pH-like BLL is high risk, typically in high-risk leukemia. So it's relatively rare to see pH-like associated abnormalities in standard risk patients. But of course, it can happen. And this patient, regardless, did very well and was in remission after his initial treatment. Our second novel fusion was another JAK2 fusion, and here optical mapping showed evidence of an inversion on chromosome 9, again with breakpoints with JAK2 and this novel gene STRBP. RNA-seq showed evidence that the fusion was expressed and that it was in frame and had entire JAK2 kinase domain. And similarly to the previous case, we were able to confirm that this fusion was the key driver because the case did classify as pH-like, as you would expect from BLL that is driven by JAK2 fusion. However, clinically, this was a completely different story. The patient here was 23-year-old with relapsed BLL that relapsed after bone marrow transplant and also after CAR-T treatment. So this patient had very aggressive leukemia. And by the time we finished characterizing this fusion, unfortunately, was already published. Another case was published by a Japanese group. But what was interesting to us was that this was another very high risk or aggressive leukemia case. So their patient also relapsed after transplant and CAR-T treatment and did very poorly, suggesting that JAK2 fusions are not all the same. It has been hypothesized that the fusion partner will really make a difference and that you have to detect the exact partner to be able to have good prognosis. But we, for most of these fusions, have very few cases, so we cannot still tell how the fusion partner will affect prognosis. Here is the third case of a novel fusion. So again, from optical mapping, we see evidence of a translocation between chromosomes 9 and 11 with breakpoints in PAX5 and the novel partner HIPK3. The fusion was expressed based on RNA-seq data, and as expected, this leukemia clustered with other PAX5 alt cases, and that's what you would expect for leukemia that's driven by PAX5 fusion. And then finally, we had a novel ETV6 fusion with a partnered MFN2, and it was due to translocation between chromosomes 1 and ETV6, 1 and 12. And the fusion was expressed, and this leukemia clustered as with other ETV6 RUNX1 cases. So we classified as a ETV6 RUNX1-like. To conclude for the cases where we had both optical mapping and RNA-seq data, and when we had both methods, optical mapping showed chromosomal rearrangement and showed chromosomal mechanism that generated abnormal fusion. RNA-seq analysis confirmed that the fusion was indeed expressed and allow us to identify genetic subtype. And having for novel fusions, two totally independent ways of confirming them, one DNA-based and one RNA-based, was very reassuring that these were real findings. We did have some cases where we did not have concordance between two methods, where only one method was able to detect the key driver. And here is an example of a case that was only detected by RNA-seq analysis. So this was EPOR upregulated case, a case with upregulation of EPOR. And this is typically due to insertion of EPOR gene, truncated copy of EPOR gene into IGH locus. So as I mentioned earlier, optical mapping cannot resolve this, but it was picked up easily by RNA-seq. The case clustered with other PH-like cases. And then what you can see by RNA-seq is a very high expression of EPOR, but only for the portion of the gene that is retained and inserted into IGH locus. And then the portion that's truncated, and you have zero expression. So this is a very characteristic pattern that you see in these cases by looking at RNA-seq data, and it's different from normal cases where you have evidence of expression for the entire gene. And this is a case that was picked up by optical mapping only and could not be picked up by RNA-seq. Optical mapping showed evidence of translocation between IGH locus and the locus on chromosome 11 that has some microRNAs. So hypothesis is that this abnormality results of an upregulation of microRNAs in this cluster. And this was described as a very rare abnormality in BLL and could not be picked up by expression profiling from RNA-seq data, probably because there are so few cases that this was not recognized and not present in the control cases as one of the recognizable expression profile. So in summary, from our experience so far with these technologies, we believe that both optical mapping and RNA-seq analysis are promising comprehensive assays for BLL. Both assays showed excellent concordance with our clinical diagnostic testing. But they also had additional diagnostic yield in unknown cases, about 30% for optical mapping, and then 78% of unknown cases were classified by comprehensive RNA-seq analysis. For our novel fusions, optical mapping and RNA-seq had complementary role in their characterization. And then finally, we are very encouraged by these results. So we are continuing with running our cases prospectively, so using both technologies prospectively on our current cases, and we will be validating them for clinical use. So with this, I would like to thank you for invitation to join you this morning. Thank you for joining. I also want to thank all my colleagues, faculty, and staff at the Center for Personalized Medicine at CHLA. And I will be happy to take questions.
Gordana, this is Leo Firk. I'm the department head. That was an absolutely spectacular talk and a great deal of information. A couple of practical things. What's the throughput, turnaround time, or whatever for the OGM? One and two, do you see, I mean, you know a lot about this. Do you see that replacing much of or a good part of the clinical testing you're doing now?
Yeah, both excellent questions. So we find in terms of throughput and turnaround time that it's very applicable test for a mid-sized clinical cytogenetic lab. So you can run 6 samples simultaneously, but you also do not have to run them all at the same time. So you can either run batches of 6 samples, but you can also run them non-synchronously, if I can put it that way. And I would say that it takes about 3 days to finish it from beginning to end with DNA isolation, labeling, running, and analysis. It can be done in 3 days. So in terms of the volume or throughput and turnaround time, I think it's very well suited for a mid-sized cytogenetic lab. I think that they're working on high throughput instruments, something that will be more suited for large scale, really big commercial laboratories. In terms of perspective of this replacing other cytogenetic methods, that's very hard to tell. I think that in terms of having characteristics that it needs to be a very good clinical test, it does have it. And so we just talked about throughput and turnaround time, and in terms of labor cost, ease to use, robustness, it meets all those marks. But it's very difficult to predict how things play out in the clinical realm.
It looks very promising.
Looks very promising. That's a very good way to put it.
Thank you.
This is John Cross, and thank you so much for that fascinating talk. I just wanted to ask, to what extent have you been able to translate this data directly into changes in treatment for particular patients?
Yeah, so it is a retrospective study. So if we had this data at the time of a diagnosis for these patients, definitely would have made a difference. Many of the abnormalities that were detected, I wanted to just show some examples of those. And so many of these subtypes, we know their risk. They belong to some subtypes that there are clinical trials for some targeting agents. So the JAK2 fusions that I show, and there are trials through COG to use kinase inhibitors in addition to chemotherapy for these patients, and we really believe that that will improve outcome. And for other detected abnormalities, they are prognostic markers. So all the MEF2D fusions would have been associated with high risk. And the NATM1 fusion is a low risk fusion in infants, which is unusual because usually infants have very high risk leukemia. And so many of these are already known as prognostic markers. Their prognostic significance is well characterized, or some even have targeting agents and that are still not standard of care to be used, but are being investigated in clinical trials. This PH-like case with PDGFR beta fusion, and that's Abl type kinase fusion. So for these, there are trials to add Abl type kinase inhibitors to chemotherapy. So definitely there would have been some changes in management if this was done at the time of diagnosis. And that's why I'm saying that for this disease, for BLL, it is really important to be able to detect genetic underpinnings and genetic driver in as many cases as possible, because it's such a well-studied disease that for many of those, most of those, we already know what are their clinical implications. So you want to really go down to the last case if you can.
So Gordana, this is Betsy. So that was a terrific talk. And I have a question with respect to, so let's say in the STRBP JAK2 fusion case that you had, when you look at that, the OGT, there were also a number of other fusions that were present. So how much, I mean, you kind of picked the driver and then tested that. How much of the other types of fusions that you see or secondary rearrangements with bionano or whatever optical gene mapping technique you're using, do you actually have to sort through? Is that a significant problem or an insignificant problem, really? You're just collecting so much data. It's terrific. But the question is, I mean, with RNA-seq, you're able to test each of those probably to see if they're expressed. But I'm just wondering how you approach that.
That's an excellent question. And this is really another reason why we were so happy to also have RNA-seq data for these cases, because as Betsy nicely pointed out, with optical mapping, we see many more rearrangements than what we expected or what we saw by cytogenetics and FISH in those cases. And so then the question becomes, so what is the significance of all these other findings? And I have to say we're still learning. So I don't have a good answer today. Yeah, many of those are even might end up being recurrent. I shouldn't say. Well, maybe I can say false positives that can be filtered out over time as we collect more cases. Similar to what happened with arrays, every platform has some recurrent artifacts that you learn and you filter out. Some of them are real, but probably not having a functional and clinical significance. And so I think with time we will manage them much better. Now it can feel a little bit overwhelming. There are additional findings that we don't exactly know and what their significance is. But it is an excellent question. However, when you think about clinical use, you can streamline it and maybe focus on a known driver. And then for others, if you do not have evidence that a known oncogene or known cancer-related gene is affected, you may just report them or document them, but assume that they are not contributing, or we don't know how they might be contributing.
Betsy, you're on mute. You're on mute, Betsy. My question actually follows up from Dr. Hirsch's question, because we are doing a project with optical genome mapping, and we've run about 3 samples thus far, and trying to analyze them and trying to optimize the filtering strategy is a little bit challenging. And I was wondering how long or what were some of your strategies for doing that filtering of results?
Yeah, so we start with what they're recommended. What recommended filtering strategy is from bionano, but there is also collaboration ongoing between some of the users. So there is a group of laboratories that have been using these cases for heme malignancies over time, and we are trying to put together some recommendations for filtering strategies for QC evaluation, for interpretation of those additional findings that Betsy referred to, because we all struggle with the same issues. So we have collaborators from Europe where this technology has been used for a longer time and much more than in US, even clinically, and then several laboratories here in states are involved, but we are collaborating on putting out there some guidelines for filtering, for QC evaluation, interpretation of additional findings, and some of the additional findings, as Betsy mentioned, if you evaluate QC for those, you can exclude them as probably not being actual calls. And so right now we are working with others to come up with some recommendations about that.
Great, thank you.
Okay, we're going to close up today because we have a 9 o'clock. Thank you, Dr. Orkana, for joining us early this morning, and Betsy. We will let you go. If anyone has outstanding questions, we do have the information to contact our guests today. Thank you much.
Thank you very much for the invitation. It's really a pleasure.